It is my great pleasure to introduce Dr. Anna Greka. Dr. Greka is a core member and director of the Kidney Disease Initiative at the Broad Institute of MIT and Harvard, where she serves on the Institute's executive leadership team. She was also a professor at Harvard Medical School and a physician in the Department of Medicine at Mass General Brigham. And we at the foundation are just so grateful to Anna and her team for all that they have done in researching the biological pathway, which I'm sure she'll talk more about today, in really setting the groundwork for finding treatments. And so we're just grateful for her team's work. Um, she also founded in 2024, I believe, the Ladders to Cure Accelerator, and this is to accelerate advances leading to treatments and cures for patients with all different kinds of genetic diseases, given the research on what she calls the nodal biological pathways. Um, so anyway, without any further ado, I just want to get right to Anna's presentation. And again, thank you, Anna, for joining us today.
Thank you so much, Sharon. And thank you to everyone at the foundation and all the colleagues who are here today. It's really an honor and a great pleasure to be with everyone and to be a part of this summit. And I just want to take a moment to acknowledge just how amazing it is that we're all here together for these next couple of days. It's not so long ago that, there was just a handful of us. And now this community has just grown by leaps and bounds. And it's just so wonderful to see that because it really takes a village to get to where we want to go. And so I'm just really so very pleased to be a part of all of this. These are my disclosures. It is relevant that I will speak about our collaboration with Alexion and AstraZeneca as part of our discussion today. Um, so I just want to start with the idea that we're actually living in what I call a golden era in biomedicine. We have the ultimate goal of preventing, curing, or treating, managing all diseases for all patients. Of course, as a physician scientist, this is my ultimate goal for my career. Of course, I'm slightly partial to ADTKD and all of you patients, with these diseases and just trying to make headway in this space. But I just want to acknowledge the fact that, obviously, I liken our work to climbing a mountain because it is a very long and arduous process. Sometimes you go up the trail, sometimes you have to stop, sometimes you have to backtrack and make your way back up again with new plans. And I think many of you have followed this path for a long time, know that it's not a straight shot, to where we want to go. And yet, we have never had more tools available at our disposal in order to get there. And so I just want to say that, we have small molecules or pills, if you will, that allow us to move forward in this area. And I think we've made headway there. We have next generation treatments, including, for example, ASOs, antisense oligonucleotides. I will speak very briefly about that today. So we have additional backups, if you will, additional strategies. And there is an opportunity for a cure, which would mean to go inside the kidney, in the kidney cells, in the nuclei of those kidney cells and fix the DNA misspelling that causes these diseases. We are far away from that. I just want to make sure that everyone hears me say that. But nevertheless, it's not science fiction. We have the technologies to be able to do that. It just will take some time before we can, safely deliver them and efficiently deliver them to the kidney. But we do have opportunities that go from managing the disease with the pill all the way to a cure. And I think that places us in this golden era in biomedicine where we have all these opportunities available for us and for the next generation, if you will, which I know many of us here care deeply about. My group is interested in membrane proteins. We're interested in human genetics and membrane proteins and how disruptions in these proteins lead to disease. And I'm just showing you here, in addition to MUC1 and UMOD, which of course everyone here understands and recognizes, some of our prior work has also been in membrane proteins. And the reason I say this is just to create some reason to believe, if you will, that we can harness our understanding of these diseases in order to develop treatments and cures. And so just as an example, my group discovered the work that went into targeting the TRPC5 ion channel for a different set of diseases. And we reached all the way to phase two trials. So phase one is safety, phase two is efficacy. We reached phase two trials in patients with that discovery from our lab. And this is now a project that is with Bristol Myers Squibb, and we'll see if it can make it to the pivotal trial. But I'm saying this to say, while this is not related to ADTKD, that we have experience bringing treatments all the way to the clinic. And this is exactly what we're aiming to do here. Even if the path is not straight, we are absolutely determined to get there, and we have done it before. MUC1 and UMOD ADTKD diseases. Okay, so how do we get there? I just wanted to present a little bit of background as to what motivates this concept of nodal biology that Sharon briefly referred to, and this founding of the Ladders to Cures Accelerator, because it's relevant, very much relevant to ADTKD, and I would love all of you to be a part of this idea. So the first thing to say is the discovery and development of new treatments is a risky process and it's difficult. It's again like climbing up the mountain, as I said. And I hope that it wouldn't surprise most of you to learn that, unfortunately, even today in 2025, the likelihood of going from a first in human, so from the phase one, the first in human study, all the way to approval remains at 13%. This is probably one of the most failure prone human enterprises that one can imagine, drug development, right? So it's a very long and difficult process. And even with all the tools, this golden era that I just made the case for, we still have a long way to go. And there are, of course, more and more tools coming in line that are available to us. But I just want us to maintain a level of humility about the task that is ahead of us. We want to get all the way to approval. The odds are not in our favor, and we have to take multiple shots on goal in order to get there. So there are many reasons for these failures, but one of the reasons that I would like to discuss with you today is that our traditional approach of taking one disease, identifying one target, and then developing a singular drug for that target, this linear approach, if you will, is one that, of course, makes it difficult for us to get to success, because if we fail to make the right prediction at any point along this path, this singular path, I think that gives us fewer shots on goal for the different diseases that we are targeting. So what I would like to present, and obviously Albert Einstein said, in the middle of difficulty lies great opportunity. I see this as an opportunity. Someone could say, oh my God, these odds are so disheartening. What can we do? But I do think that we have an opportunity. And the opportunity, one of the opportunities that our team has spearheaded is this concept of a biological node, of nodal biology. So a biological node, we define it as a point of convergence on a single druggable node, the druggable target, that enables the discovery of connections, previously unseen connections between diseases that may have nothing to do with one another. And yet now they're connected because they share an underlying mechanism. So we're not going by symptoms or organs, but we're going by biology, the underlying fundamental biology. And of course, with the advent of artificial intelligence, we can actually use these tools to identify more nodes. And that's part of the mission of the accelerator that we have built at the Broad. But all of it was inspired by ADTKD. And the idea that we found that there's a shared druggable node that is not only shared by MUC1 and UMOD, but by many other proteinopathies. These are diseases that are caused by misshapen, mangled proteins that accumulate inside cells, but not only in cells in the kidney. They also accumulate in cells in the eye, for example. And so we're also working on a form of blindness that runs in families, just like kidney disease runs in ADTKD families. And we are also working on a form of Alzheimer's disease that runs in families and other diseases that are also similarly genetically defined proteinopathies. And this TMED cargo receptor is a node that can serve all of these diseases in a way that is completely unpredictable. No one would have thought that an eye disease and a kidney disease would have something in common, and yet they share this same underlying mechanism. And so we have the opportunity to develop new drugs, not necessarily a singular drug, but multiple drugs for all these different diseases, but by targeting the same molecular node, which actually now gives us more shots on goal for all of these diseases, right? So this is the concept of nodal biology that I wanted to introduce. In the case of ADTKD, again, with the advent of artificial intelligence or machine learning, this is a structure that was predicted by the computer for this druggable node, the cargo receptor node that I just showed you. What is in orange is a particular component of this node that we are very, very interested in because it's an opportunity for us to develop a very specific drug for this node. But I just want to make the comment that by the combination of artificial intelligence and all of our prior understanding of the biology, we now have these incredible tools that allow us to build bespoke drugs, if you will, that just so will fit into the right place in this structure that you see. To you, it may seem like ribbons dancing. For those who are the scientists here, it will make perfect sense. It is the structure, it's the multimeric structure of a thymic cargo receptor complex. But the point is that this alpha fold structure gives us an opportunity to develop the right drug for this target. And as many of again, I mentioned to you, drug development is a very risky process. The more information you have, as we now have, the better drug we can build. And so, we have a new compound that fits just so into this pocket. We understand much better how to deal with this. This is information that allows us, to move this project forward and avoid prior, liabilities or toxicities, if you will, that these small molecules may have had, so that we can actually build better compounds. So all of this knowledge, including the structural information that we now have, allows us to build better drugs. And so I just wanted to show you using not the new compound, which I can't say much more about, but more to say that we're using the tool compounds that we have previously developed at the Broad. I can show you how well it works in the kidneys of animals that have MUC1 and uromodulin kidney disease to give you a reason to believe that we now have everything we need in our armamentarium to build the best possible pill. We don't have it yet, but we're building the best possible pill to address these diseases and to put it into phase 1 trials as quickly as possible. And so this in green shows the tubules of a knock-in MUC1-FS mouse. These tubules are not normal, just like in the patients. As you can see in the biopsy here, these tubules are very wide. It's called tubular dilation. If you give the drug for 3 months to a mouse that carries the human MUC1 mutation, then you see that there are these big dilated tubules shown here in this orange circle. If you give the drug, the tubules become we're able to prevent the tubules from becoming so wide, is a better way to say it. And if you look at the control animals, so these are the animals that do not have the mutation, you can easily see that the tubules of the treated animals, where we have prevented this dilation from occurring, I want to make sure that you understand that these are all shown at the same magnification. This is not a zoomed in picture. This is how big the holes are, if you will, or the dilated tubules. But this is where we're able to mitigate that from happening so that they look much more similar to normal control kidneys. So if we could do this in a human kidney and we can prevent this from happening, we would obviously have a very, very attractive treatment for this disease, right? So very much interested in advancing this knowledge toward clinical trials in patients. Now, what about UMOD? I want to make sure that we address both diseases. This is a mouse model that carries one of the UMOD mutations, as shown here. And I'm showing you here data that we published about a year and a half ago, where we basically treated these animals with the tool compound. And as you can see, we have a very significant reduction in the mutant protein in these animals. This is with a mutant-specific antibody that we were able to raise for this mutation. And we're also able to reduce the target of the drug, the cargo receptors that we're looking at. So we're reducing both the misshapen protein and the cargo receptors that hold on to it inside the kidneys. And so we're able to mitigate the disease in the case of UMOD as well, using the same nodal approach, right? This is showing us that we also have the opportunity for a tremendous biomarker, very much building on Martina's brilliant work and the work of many others who have collaboratively worked for many years in this space. But the idea is that, with the treatment with the drug, with the tool compound that is sort of the inspiration for the future drug, we can actually detect an increase in uromodulin in the urine of these animals, which again suggests that we're restoring the kidney function in these animals. And we also have a readout for when we are doing that. So if we can measure something in the urine that tells us that the drug is working, this is a huge advantage for being able to take our program toward the clinic. And so I'm just showing you here some of the we call this biomarker development that one can do for this disease. Silvana, who gave a talk at this summit last year from our group, presented some of these data, but I just want to remind everyone that she has done a brilliant job in this study in collaboration with Martina and Stan and many others to actually take tubules from the kidneys of these animals and isolate them and then test the effects of the drug in these tubules. And as you can see, the red in the middle is where the uromodulin is not supposed to be. It's accumulating, aggregating inside these tubules in a way that is not good for the tubules. It's not functional. However, if you treat the tubules with the drug, you can see that the uromodulin becomes normal. This is what it's supposed to look like, to have this linear pattern on the surface of the tubules. And you see this also better here, which basically means, again, that we're able to restore the normal uromodulin where it needs to be, while hopefully getting rid, as you can see, of all the bad uromodulin that is accumulating inside the tubules. So very nice results here as well to show that the drug is working in this setting. And so, to summarize, essentially, we have developed an understanding, a much deeper understanding of this nodal target, the TMED cargo receptor target. And as I mentioned to you, we are now doing work in retinitis pigmentosa and a couple of other proteinopathies as well. I won't show you the data, but just to tell you that this is actually an advantage for ADTKD in the sense that you can actually group this disease together with others because that can increase interest in this therapeutic strategy and it can bring more attention to this program, if you will, and potentially more investment as we move forward, because it allows us to understand biology that is relevant to many more diseases than just ADTKD. So I want you to all see this as especially for the patient side, to see this not as us being unfocused, but actually being doggedly focused on finding a solution to this disease by taking this nodal approach that allows us to increase the applicability of this biology and this program to many other diseases. So it is an advantage to be able to take this nodal approach for this disease. Now, you could say, well, what if any of your efforts with the small molecule don't work? You just told us that there's a lot of opportunity for failure. And I'm, again, continually humbled by the complexities of drug development. So as an alternative, we're not sitting on our hands. As an alternative to the small molecule compound, we actually are taking advantage of a different technology, which is called antisense oligonucleotides, or ASOs. This would be an injection in the arm. It's otherwise known as a nucleic acid therapy. It wouldn't be a pill that you take, but it would be an injection in the arm. And just for a brief reminder for those who may not know what ASO technology looks like, I'm taking as an example the MUC1 mRNA. So this is a kidney cell. This is the nucleus of that cell. And here's the MUC1 mRNA. So the message that Martina so elegantly explained to you, from the DNA, we get to the message RNA. And this is the ASO drug. So it's a small sequence that is complementary to the mRNA. And then we provide it to the cells. The cells take it in, the kidney cells take it in, and the ASO goes into the nucleus of the cell and it finds its complementary mRNA, binds it. And that binding event, it turns out, when you have a good ASO, results in the degradation of the mRNA. So we are preventing the mutant protein from being produced. The protein is not even made because we are getting rid of the mRNA from MUC1 in this example. One can do this for any other gene. And so the idea is that we're able to develop a completely different approach unrelated to the biology that I previously shared with you with the TMED cargo receptors. It's a completely different approach that allows us to essentially get rid of the mutant mRNA in this case, right? So this is a different approach to this. And I'm just showing you that we have actually been able to successfully develop an ASO. In this case, I'm showing you an ASO for MUC1, but we're working hard on additional approaches for UMOD as well. And this is an ASO that was given for 3 months, for 6 weeks, actually, to this mouse. And we have not seen any toxicity, but we have seen more than a 75% reduction in the mutant protein as a result of using this ASO. So we have a very impressive reduction of the MUC1 in the kidneys of these mice. Again, we're only curing mice so far, but we're trying to get there for pushing this project forward toward humans as well. And one of the key points here is that there's no toxicity because there's been a lot of concern that these ASOs may confer toxicity. And so we are not seeing any of these signs here, which is a very, very encouraging signal. So what this means is that we now have another shot on goal for this disease using this technology. And of course, we're exploring all of the nucleic acid technologies that we might be able to use to treat these diseases. And so I'm just giving you this as an example to show you what we're doing. And so if I were to summarize today where we stand, we are very much pursuing our TMED targeting compound, the pill, if you will. Ultimately, it would be an oral pill that we can take for these diseases, which would be both for MUC1 and UMOD. And we're also pursuing early days, but we're pursuing ASOs and other nucleic acid therapies that would be delivered as an injection in this case. But those would also be therapies that can really, hopefully, as we see in the mouse models, adequately address this disease. And so I just want to end by where I began and to basically say that we really do live in an era where we have more tools at our disposal than ever before. I am very determined that we will do everything we can to get there in terms of the ultimate goal at the summit. I am humbled by the complexity and the difficulty of the path, but not daunted. I think that we have every opportunity to continue to fight to get up to the top. And I have no doubt that together, working together, and I think this is really important as well, that it takes a lot of stakeholders working together, including the patients and the families, all of us together to be able to reach our ultimate goal. And so, on behalf of a very grateful team at the Broad Institute. I want to thank all of you for your attention today and for this opportunity to share with you some of our recent progress. As you can see, it also takes a village to get all this work done. And I'm very lucky to have an incredible team of scientists working with me. And also an incredible team of collaborators. You've heard from Martina. We work so closely with Tony, with Stan, Kendra, and Greg also in Cyprus. And, we couldn't be luckier for all the other collaborators. You heard about Peter Conlon and many other investigators all over the world who are working together with us in every country, really, to be able to build, the registry, the understanding of the disease, and ultimately the therapies that will allow us to get there. And so I encourage all of you to always stay in touch with us. We are always inspired by collaborating with patients. And again, I thank you very, very much for this invitation, and we'll be happy to take any questions for any time that we might have. Thank you so much.
Thank you so much, Anna. I'm just sitting here. I have goosebumps. I'm just so
Oh.
excited to hear where you're going, the efforts, and your commitment, your undying commitment, and optimism is just, it's making my day and my life because I think of what this
Thank you.
means for my sons who have yet to be tested, but odds are at least one of them has this. And I don't want them to experience what my parents, my families have. And I'm speechless. So just thank you so much for being here and being such a valued partner in all of this.
Thank you. That's wonderful.
Um, Let me turn it over to Lisa and see. Let's see, we've got about 10 minutes for questions.
Oh,
What
good.
questions do
Yeah.
we have for Anna?
Good morning, Dr. Greka. Let's see here. Okay, we've got a couple of questions. First question is, earlier publication claimed BRD 4780 to be the chosen compound for MUC1 dependent ADTKD.
Mhm.
What was the reason for testing BRD 7635 for MUC1 when TMED is central protein for both MUC1 and UMOD?
Yes, all of these are tool compounds. So they're all, 7635 is an improved version of 4780. And we continue to make improvements as we understand more and more about the target. As I mentioned, now we are structurally enabled to some degree. This is an AlphaFold structure, but it's based on the cryo-EM structure that we published a couple of years ago for TMED9 homomeric. structures. And so, as we understand more about the target, we can build better and better compounds. So 4780 and 7635, I want everyone to understand at this moment, they are tool compounds. They will never become drugs that go into humans. They are just tools that we are using to understand the disease. We initially thought that they might have legs to go into the clinic, but that has since proven not to be true. But we are now on the road of building better and better small molecules that will eventually, one of them will be selected to go to the clinic. And we're closer than ever to that. But it's a process. And, this is what I mean that like when you're climbing up the mountain, sometimes you think you're getting somewhere, but then you have to stop and backtrack because, you discover that there's a toxicity or a liability with some of these compounds. And then you have to design new compounds where you're selecting away from that liability and more towards specificity for the target that you're going for. So I hope that this helps answer the question, but we're getting smarter and smarter about developing an ideal small molecule that can hit this target. And these compounds are just on our way to get there, essentially, right? Like the iterative process of getting there.
Wonderful. Okay, next question. CRISPR research for ADTKD. Any update?
Yes. So we are also working on CRISPR technologies for those who are familiar, this would be opportunities to actually cure the disease. So it would be a one and done approach using CRISPR technology or other such molecular machines like CRISPR, for example, base editors or prime editors that were discovered by my colleague David Liu at the Broad. We are very much working on these approaches for both MUC1 and UMOD. However, there are many hurdles to get there because nobody right now knows how to efficiently deliver these kinds of molecular machines into the proper cells in the kidney. So we are continuing to do a lot of discovery work in that area in collaboration with the Liu Lab at the Broad, who are the world's pioneers. So for anyone who saw, the best, perhaps the best example, the most beautiful example of what CRISPR can do is baby KJ, who was in the news last year, this little baby in Pennsylvania who was treated with a base editor in the liver. So the delivery to the liver is solved, and it's something where we can actually much more readily deliver these tools. And yet it's even early days for that. So the kidney is behind because we haven't really solved the delivery problem. And so we have much work to do, but we are doing it. So I want to make sure that you understand that that's an approach that will take much longer, but we are very much determined to work on that as well until we can solve it. So very much ongoing work. I didn't speak about it today because there's not much to say in terms of updates. It's all sort of boring technical details that we're working on, but we are working on it, very much so.
Okay, we've got one more question here. I'm guessing this falls under the heading of things that are humbling about the drug development experience. But can you speak a bit about whether funding is or has been a challenge along the way, especially given these diseases are rare?
Mm-hmm. Yes, absolutely. I mean, there's always this issue of, this is part of the reason why we started the accelerator at the Broad, because it is often the case that these diseases are relatively neglected in the drug development process. Again, especially since I mentioned to you how perilous the process is. So if the patient population is small, you can imagine that doesn't motivate a lot of people to work on this problem. However, genetic diseases, also rare genetic diseases, have their advantages. In this case, we can easily identify the patients using genetics, which is why it becomes of paramount importance that this organization exists and that this summit exists every year. We need to raise awareness so we can find more and more patients so that when we are ready for clinical trials, we have a patient population that is ready. This is very attractive to those who make investments into this space. So the companies that build clinical trials for rare diseases want to see a patient population and clinicians who work with these patients so that they can execute on these clinical trials as efficiently as possible. So this foundation that we're all building here together for the next 2 days is crucial for being able to get these new potential therapies over and into the clinic. And so everybody needs to come together, the clinicians, the scientists, the patients, the families, the foundation, the regulators. It truly takes a village. All the investigators have to work together so that we can build the necessary foundation for clinical trials to be executed as efficiently and as quickly as possible. So funding is always a challenge. I'm constantly fundraising for rare genetic diseases, including ADTKD. We have been blessed with funding so far for all of our different scientific projects. And we want to make the best of this truly a gift that is bestowed on us, the gift of funding, to make the best of this resource so that we can actually get as many shots on goal as possible and build as many therapies as possible that we can advance toward the clinic. So, and go as fast as we can, of course, always maintaining, the protocols and safety and all these things that need to happen in order for treatments to make it from mice in the lab to patients ultimately. So thank you for your question. It's an ongoing struggle, but one that I welcome because I have a great sense of sort of purpose and meaning from this work. And I think I speak for our entire team when I say that we feel a great sense of purpose in doing this work on the benefit of patients everywhere.
Thank you, Anna. We have couple more minutes, and I'd like to just ask you to take a step back. Where do you see this field in, say, 3 to 5 years, 10 to 15 years? I mean, understanding that, you can't predict and it's a
Yeah.
mountain and ups and downs, but give us a vision for at least what you think is feasible and what you're working on.
Sure. I mean, yes, it's always difficult to make predictions, of course, and as I mentioned throughout my talk, I've been humbled many times, and so there's always the possibility that we try to go up the trail and then we have to backtrack and regroup. But I think over the next 3 to 5 years, I predict that we will have one or two clinical trials starting in this space. I think that's a very realistic goal. I think that we have every reason to believe that the small molecule program and potentially the ASO program will make it toward the clinic. I'm very bullish on that. I think that we have a good chance to at least take our best shot, right? Like that's all we can ever do, and hope for the best. And I do think that over the next decade, we are going to make substantial progress toward the gene therapy goal, because ultimately what I'd like to see maybe toward the end of the arc of my career, I would love to be able to cure these diseases, right? So this is not something that we will be able to do in the next 3 to 5 years. I think that will be for the kinds of treatments that manage the disease, like the pills, the ASOs, these kinds of things. But then for a cure using CRISPR, base editors, prime editors, these kinds of technologies, maybe the 10 to 15 year horizon seems more realistic. But I think that that's also very hopeful. And I speak of that as a physician, as a scientist, and also as a mother myself, right? I mean, I would love for these treatments to be available so that the next generation doesn't have to worry about, what our colleagues and patient friends mentioned earlier. Cliff, for example, this worry and stress about sort of managing chronic genetic disease. Hopefully, the next generation wouldn't have to suffer from that. But, well, we have a lot of work to do in order to be able to do that. So, and again, I may be humbled, and please, nobody here hold me to the timelines. I know this is recorded, and you can bring it back in a few years and say, Anna, you said, all I can say is that everybody here will do the best we all can, right, to meet those timelines or even accelerate them as much as possible. But we do need to reach the clinic over the next, certainly 3 to 5 years yeah, I think that's a realistic goal. And then for developing, the kinds of tools that would lead us to a cure, I think that's a longer horizon, a 10 to 15 year horizon.
Thank you so much. That's helpful. And we promise not to hold you to those numbers, but to
Oh,
just
thank
to
you.
give people a sense of time in all of
Right.
this, given the
Yeah.
complexity it takes.
Yeah, I mean, this is what I want to convey to everyone. the fact that I'm not, I'm saying, don't hold me to the time is not because of any lack of determination or commitment, but it's because with all of that, you can get to an unpredictable sort of point where you have to backtrack and then you're delayed. And we've already seen some of this happen in these programs, but that doesn't deter us from continuing to move forward, learning, regrouping, and being able to bring the project forward. And so ultimately, this is what it takes. If you look at any other, for example, I invite everyone here, I'm sure many of the scientists know this. If you look at the revolution that happened with cystic fibrosis, the patients, the foundation, and the scientists, the physician scientists, worked for years in order to be able to bring these therapies that now make that disease. I mean, they have totally revolutionized the outcome for that disease, right? And so these patients, these children with cystic fibrosis are now, living basically a normal, life, right, with those treatments. But what did it take to get there? There were so many twists and turns along the way. And, if you ask seasoned drug developers, they will tell you that every drug development program dies at least 3 times before it's actually resurrected to make it all the way to the finish line. And so we just have to be determined and, continue to fight and move forward, understanding that there's twists and turns along the path. So as long as we're all united in that understanding and we work together, I have no doubt that we will ultimately get there. I mean, our being together today is a testament to that. But at the same time, we have to be hardworking, realistic, resilient to take the setbacks and then continue to move forward with determination. So that's my message for today.
Which is greatly appreciated. And your and your team's commitment and persistence is just palpable. And so we just
Thank
are
you.
so grateful for that. So thank you for joining us
Thank you
today.
so
I know
much.
you need to take off, and we'll be in touch. Best to you.
Thank you. Thank you. Greetings to all.